You, me, and HIFMB. Stories of Science and the Sea.
Hey everybody, and welcome back to the HIFMB podcast. We are nearing the summer break, which is probably what you saw in the very infrequent release dates of the podcast episodes lately. And it's been getting crazy busy for me, so I'm in between projects where I'm still wrapping up my old project, but I'm already working in the next project, so it's a bit of overlap for me, and I try to find time to record podcasts in between, which is very few and far between these days. But we have a really cool podcast special, a little series coming up, but I can't tell you too much yet. So your waiting in between episodes lately will be rewarded. You can also hear that we're nearing the summer break because in this episode we had so much thunder and rains in the background, and it's a good summer episode for you. To make your summer more interesting, this episode is by Anna Roik, a postdoc who works on coral reefs. What she does on coral reefs is she actually works on the microbiome of corals. So that is harnessing the microbes that actually live on corals and provide a lot of benefits for them. And she pioneered, with some other scientists, the method of transplanting that microbiome from one coral that seems to be very resistant to something like global warming, where normally a coral would bleach. These corals, they seem to be quite resistant, and Anna and her colleagues, they harness the microbiome and transplant it to a coral that doesn't display these heat-resistant characteristics and actually makes it more resilient to global warming. We also talk about her career and how it's quite logically followed from one topic to the next, and specifically dive into her time in Saudi Arabia, which is quite an interesting topic. So without further ado, I give you Anna Roik. Hey everybody, and welcome to the next episode of the HIFMB podcast. And now, fitting summer, we have marine scientist Anna Roik here, who focuses on coral reef ecosystems.
Yeah, hi Jan.
Hello.
Happy to be here.
So today, first of all, who are you? It says on your website that you are a marine scientist with the focus on coral reef ecosystems and the microbiome of corals.
Yeah, so that's pretty much it. So I work on corals and microbes, and to keep it really short. But so technically speaking, if I want to introduce myself and want to explain what I do, yes, I'm a marine scientist, and my major focus, especially at this moment, is trying to develop interventions and adaptation strategies for marine wildlife in the face of climate change and ocean warming.
Okay.
And to do this, we basically focus on the like a marine organism, which is in my case the coral, because coral, like I have been sticking around with corals for a while, so I know these organisms already pretty well, I would say. And the other part in this quest is the microbiome. So the microbiome is like an associated community of tiny little unicellular organisms. And these organisms are actually, we don't know much about them, and that's why I integrated them at some point into the scope of my research, because we really want to learn about them in the end. So we know that a microbiome can be powerful, and we know it from us humans, from like medical science that has been advancing.
Like our gut microbiome.
Exactly.
Like that,
and so we base everything I do is based on the hypothesis that the microbiome could be harnessed and could be used or manipulated to do good for the coral, which is endangered, as we know, especially by climate change. And it's warming. So the major problem is warming that we're looking at.
Yeah. Okay. And backing up a little bit, like you said, so you first, how did it come about for you? Did you first focus on corals and then focus on the microbiome, or the other way around? Like you always focused on microbiome and then used corals as an example, or?
Yeah, so that it started with the corals, I would say. So I was working, so my very first job on the reef was working in a restoration project. So I was involved in a restoration project in the Caribbean where the big goal was to restore populations of Acropora palmata, which is a critically threatened coral species, and it's I think by now I started 10 years ago, so I don't know what the status now, but it was really critical like 10 years ago, and many projects that have been focusing on saving this coral were set in Florida, in some of the Caribbean islands, where figured out that they're losing this coral, so lots of funding went into trying to breed them.
Okay.
And propagate them on the reefs.
So Acropora palmatas is this branching coral that like builds tables, right? Kind of.
Yeah, it's the Elkhorn coral. It's
Elkhorn,
so it's like it's really massive, but it's a branching coral, and it's very unique. So you don't find this type of coral in the entire of Indian Ocean. There you will find other Acropora species, but in the Caribbean it's a very iconic coral as well. So there is 2 Acropora species only, and they're branching corals, and these branching corals are really important for habitat formation. So because they're built this 3-dimensional structure, and that's why they're really important. And now they were the most. It's lightning here.
Yeah, there's a lightning on site.
Yeah, it's so a tropical storm and all that.
Yeah.
Oh.
There we go. That's new.
I hope. I'm not sure if you can hear it.
Yeah, I hope so.
We will see.
Yeah.
Yeah. So these corals are very important, and we only have two species of them for like building this 3-dimensional structure, which is critical for the maintenance of biodiversity in these ecosystems.
Yeah, like fish can hide in between the little coral branches and stuff. Yeah.
And like all the critters attached to that are kind of associated with the reefs.
Yeah, exactly.
So it's a breeding ground, and it's the habitat. So this is where I started out, and my further career and connection to corals went into doing a PhD at some point, because I really got interested into the ecology and how things work, and why for instance we would set out some corals and some of them will die, others not. So this is something to find out, I thought. So I started working for my PhD to earn a PhD title. Yeah. And I joined a genomics lab, or I had the opportunity. I applied for this job, of course. That was based in Saudi Arabia, so a different system, the Red Sea, which means for a coral reef scientist, Indo-Pacific corals, which are more diverse. So you need to learn many more species if you are interested also in like the macro community structure and so on. And then also having my genomics buddies on my hand, I got many opportunities to look into other things than like the restoration projects gave me the opportunities. And this is actually where we started looking at like some symbionts. So maybe a little bit of coral education. Corals are very much dependent on their unicellular symbionts, which are sometimes called algae, but they're not really algae. They're what we call dinoflagellate. We biologists, we call them dinoflagellates. But these unicellular organisms, they do photosynthesis, and this is how they provide energy to the coral. In this, based on this, the coral is like a symbiotic animal, and that's why we find them in these clear tropical waters mainly, because this is where the symbiont can perform at its best and receives a lot of or enough sunlight to provide the energy. At some point we started looking at the microbes that were associated, so bacterial microbes, because there's many types of microbes. Right. I don't know if you want me to go into this.
No, I just wanted to briefly ask, like you mentioned the microbiome and the unicellular organisms, the symbionts. Are they considered part of the microbiome or not?
Yes, of course. Yeah? So they are. Yeah. So we now by now we used to say microbiome compartment. Okay. So there are many compartments of the microbiome, or many different communities, which in the end probably interact with each other. So there's many unknowns. We don't know how these things work together, but. Eventually we want to make use of our knowledge and manipulate the microbiome. Microbes could help make an organism more resilient in terms of stress. because there's microbes with antioxidant capacities. So they produce antioxidant enzymes. These enzymes could interfere or interact in the cell or in between the cell, the host cells, to quench certain stress chemicals. Let's put it in that terms. So there's different, really many different pathways that could be targeted, but it's like this that's the most obvious one: antioxidants and the stress chemicals that are being released when organisms are stressed by heat or
like when a coral bleaches, it is subjected to oxidative stress, right? And antioxidants battle that.
Exactly.
Yeah, okay.
And we don't know if it's the dinoflagellate or the coral. So
we don't know. Okay.
It's not fully clear who's stressed first.
Right.
And in some cases, it could be that it really depends on the coral species and how the stress is happening.
That's super interesting. I still learned, like when I studied, I still learned that it's always the dinoflagellate that gets stressed and releases oxidative stress molecules into the coral, and then that kicks it out.
That has been shown in a couple of studies, but there's also studies showing the dinoflagellate is fine and performing fine. There is oxidative stress increasing in the cells, and you can just see host factors going down the hill. So it's not completely resolved, but we know oxidative stress is an issue, and that's why the research community came up with the first thing that was suggested. Antioxidants. That's what we need.
Right. Okay.
for the coral.
You brought this paper today, which is called "Towards Enhancing Coral Heat Tolerance: Microbiome Transplantation Treatment Using Inoculation of Homogenized Coral Tissues."
Yeah, thanks for this reading this long. I'm sorry for this title.
No,
very
that's
technical.
Sometimes it needs to be long.
It's a technical study.
Exactly. But it is super interesting. Like the first thing you mentioned is, so what you did is you transplanted microbiomes from heat-resistant corals to less heat-resistant corals.
Exactly.
Okay. And you did that with, so it's called a CMT, coral microbiome transplantation.
Yeah.
And you did it in Postolopora and Porites corals, so no Acropora here.
No Acropora here. For the reason that was a study we performed it in partly in situ. So we use wild corals in Thailand. Yeah. And there were just in those reefs that we had access to, no Acroporas left. So this is like one sad point about also working every year where when I go to especially those places that have good access to the reefs. And I mean, Thailand is a very popular place to go for holidays, and so the coastline is very developed. The corals are, coral populations are declining. I can see it in my lifetime, in my short lifetime, or my research lifetime. So we didn't find Acroporas there, which doesn't mean they're not there. They were just not in those reefs that we were performing our work. And Postolopora and Porites. So Porites is like one of these massive brown rocks. People just many people don't know that's alive. And Postolopora is also called the raspberry coral. It's cute and nice and has little arms that look like raspberry bubbles. I don't know.
I didn't know that.
It's and it's pinkish. And most it can be brown. It varies from brown to pink. And
so you mentioned you went to Thailand with this study.
Yeah, it's on the Andaman seaside.
Like Phuket is in there.
Phuket is there, and Similan, which is maybe the most popular destination. For diving, snorkeling, but it's really tiny and it's overloaded with tourists. So please don't go there.
Okay. Holiday recommendation.
Yeah, I've never been there. I've been working mainly around Phuket and Koracha, which is south from Phuket. And let's get back to the probiotics and microbiome manipulation. So we did the study there because of personal contacts and this collaboration that we started with Thailand, and we were restricted to using certain corals because of the lack of species in the reef. And these two corals are really important reef builders at this moment there, and they're known as really critical reef building coral species in the whole of Indo-Pacific. So it's not only Thailand, it's like more than half of the marine world, I would say. And that's why the significance. So they're really important. That's why we have chosen those, because it's important to understand how we can help them and how they respond, how they, behave. We started thinking, especially in my case, I was in touch with people or with labs who have like a biobank of microbes, and they were testing potential probiotics, really administering certain marine microbes to corals in an experimental setting. I wanted to do something similar, but I didn't have a biobank, and I was in Thailand. We didn't have
I was
a microbiology lab there. So I was just I decided to go for try find a solution for also a very real world application that you can do in remote places, like or places that are maybe not well don't have the facilities for high-tech science. And that was a thing we could do there. So a microbiome transplant, we just needed to find
what do you need, like how do you do it?
So what you need is you need to know your corals. So the first thing is we developed a heat stress assay. So a totally different thing. So work on the physiology first. Figure out who. So it's like you can imagine for us humans, we go to the doctors, get a blood test done, and you will find certain parameters that are in check or not, and then this patient or this person has this gut problem and this doesn't, and this is how you figure out. Just
the
an example.
susceptible ones.
Exactly. So what we do is we test the corals, which we find in the field, using kind of a standardized assay, and we're looking at their bleaching response. Photosynthetic efficiency is a good metric because, as you already said, the microbes, the symbionts, the photosynthetic symbionts, are usually the first ones who are affected by the heat, and then you can measure it with a fluorometer, like the plant scientists use. So we use one of these tools.
Do you artificially bleach them, or are they recently bleached after a bleaching event?
So we run a heat, as we simulate acute heat stress. So we put the corals, we take them out from the reef, put them in smaller tanks. There is circulation, so they get everything they need, but the temperature is being ramped up over a short time, up to something like 33 or 34, which is above all of their, critical thresholds. It depends on the coral species. For the sturdy ones, we run it for 2 or 3 days. So we repeat this peak over 2 days, and we take measurements, and then we can figure out how fast do they bleach. And there's those that respond instantly, and there's those that don't respond at all or respond later. So we can group them in resilient types and non-resilient types. And in this area where we worked, we figured out that one island, like the western island sides, had predominantly some resilient type corals compared to the east shores that were nice and protected reefs. So that also has to do with their environmental history.
Probably.
We also have a paper on that. Because this project just gave a lot of data and lots of things to be interpreted.
Exactly. You're quite the pioneers in this field, right? With applying the microbiome to corals.
So this is the first study.
Exactly.
Then we collected those corals that performed really well in our heat stress test. We produced homogenate from the tissues. So this technique requires some coral material. So
obviously.
This is not. I mean, it's not super sustainable because we need some coral, like a donor. We need something to donate to the poor sensitive corals.
How many donors do you usually need in order to transplant so much microbiome on so many corals? Like, what are the numbers?
We don't know. We don't know yet. This is dosing. It was the first experiment. The follow-up experiment will need to figure out the dosing that still will provide the effect. So, because the nice thing was we measured a positive effect. So we didn't save the corals from bleaching, but they bleached much slower. then the placebo group just really crashed, like in the first test that we did with their when we were exploring, and trying to understand who is the donor, who's the best donor, who's the best recipient. So the recipients, the placebo group of recipients, didn't perform well at all, while our other corals made it through fairly well.
Yeah. Okay.
Yeah, but so we see some positive effects, and we could even pinpoint several bacterial species that were very likely transmitted by NGS. So we looked at them with NGS data, so sequencing data, metabarcoding, and we could find a couple of bacterial species that are already in, or some of them are already in the focus of science, of researchers.
Okay.
Because some of them we really suspect to be important for the coral organism, so potential symbionts. So we don't know yet if they're really true symbionts, but we find them, some of them we find often with corals. And by showing here that new strains or new variants, so to say, were transmitted from a donor from this western shore to the eastern shore recipients, which didn't have that, by looking at the DNA, the barcode. It was new variants of these major microbes that we suspect to be important for the coral. So that was not random stuff being transferred.
No, absolutely.
And that's also one of the outcomes that is important for the further work, because now we suggest and recommend we should focus on these potential symbionts. Yeah. Those that are
on these species that were super beneficial or that were most likely to be beneficial for the corals.
Yeah, because they're able to stick around with the host. Yeah. So there's a discussion in the paper.
Yeah, absolutely. Yeah. Very much assisted evolution, they call it, I think.
Yeah, that's also, it goes into this pigeonhole or
rabbit hole again.
There's also many different approaches to do this with symbionts or with microbes or just with a host.
So that paper came out in 2021, and you're still working on this topic. You're still involved with work.
Yeah. Well, so maybe there was a pandemic.
I was aware.
So experimental work, especially like the field work, was halted, but in the meantime, we did, as I said, we are exploring other microbiomes, like the microbiome, the fungi. I've been working on some perspective and review papers, and actually now I'm preparing for another trial that will be the follow-up to this paper.
In Thailand again, or?
No, probably in Israel at the Red Sea.
Okay.
So in Thailand, our contact, my collaborator in Thailand, went to Bangkok. She lives in Bangkok now. Right. And it's the Gulf. So Phuket. Yeah, it could be an option at some point again, but at this point, I'm starting something with a bit of a different group, but following up on the open questions that were left from this first experiment.
Excited to see what comes out of it. I
mean, me too.
And you also mentioned that aside from your research, you also like to explore the coral reefs that we see today using your camera. So you're an avid underwater photographer.
Oh, yes.
Yeah? Nice. Yeah. So you're always on like field trips and stuff? You always take your camera? And
no, that's really my free time, I would say.
Oh, right, okay.
Because on field trips, of course, I have my camera with me, but most of the time, you just don't get the chance to go to these places that you really want to capture. Because I do photography, of course, I have many photos of my field sites and some of the work that is being done at the aquaria. But it's mainly, because I told you I started diving and I really liked it, even in the Netherlands. However, since I never dove in the Netherlands again in the North Sea.
It is cold.
It is cold, and there's there's a little bit to see, but it's different. And I don't know, I really got into taking photos, and especially I have a wide angle lens. This is what you need in a reef to capture the landscape. And what's also very popular among photographers, you may also know if you go on a dive trip somewhere, you will also find these people with they're macro lenses, and they like to just sit there.
Like little nudibranchs or
yes,
something, like tiny snails.
Yeah, so we call it the critters.
Yeah.
I really enjoy doing this, and I think it's just the side effect of the places that I've been living, like I was close to the reef.
Yeah.
Close to the sea.
Nice.
And so I have to say, when I go for a walk, I also take my camera with me, or people sometimes would freak out because I'm taking photos instead of hiking, you know, when we go hiking in the mountains.
Yeah.
So it's just it's a hobby, and
Nice.
The underwater photography is pretty challenging, I have to say, because you're going on a holiday and you need to take care of your equipment all the time.
Yeah, I know.
So sometimes I'm really tired, but I'm always happy to use my camera and get the chances to go to these really special places. And because I know the reefs, so I more or less also know where to go and where not to go, and how to go there is also important. So how do you step into these ecosystems?
Yeah,
there's do's and don'ts.
Yeah. And do you then go when you've spent all week on a field trip in the sea already? Then you go on the weekend and take another dive just for fun?
Yeah, of course.
Yeah, nice.
Yeah, so that's how I started it. I mean, you need some time off. You need to relax. Yeah. And then people asked me to go on a dive. I was just really bored on a dive, so I took my camera. I think that's how it started. And by now, I think last month I went to a conference and spent 2 days on a beach nearby in Malaysia. And a friend told me, let's go diving. Yeah, but actually we went, I thought I'd go there for just hiking in the jungle, which I also really like. And I didn't bring my camera. And she had one. She had her little camera, and I was so bored in the 3 dives we did it. So this is where I figured out I need something to do in these places.
Yeah, I'm the same.
Yeah,
exactly. And then you I think that, I don't remember when it was now. I think in April you had an exhibition of your photos in Oldenburg.
Some of mine and some of others.
Yeah.
Yeah. So this was actually a nice event and an opportunity to make use of these photos. So I have some of my. I'm a lazy person actually. I have some photos on my website, but I have tons of photos on my home server. Yeah. I need a home server by now. I need to do something with that. But it happened that my colleague and I, a good friend, and also I lived with her here in Oldenburg for 2 years. She left now, unfortunately. But Stephanie Halbar, she was also working at the ICBM in Wilhelmshaven. She's a scientific diver. She was working on the North Sea projects, but in her past research life, she's been working on tropical seagrasses mostly. And she is really into photography of tiny critters in particular. So we ended up some evenings just hanging out and showing us the critters we still had on our phones from the rare weird shrimps from some island in the in the Indian Ocean. And this is how it started, because we did it a couple of times, and I think it was actually Steffi who initiated this, because she also got into creating a children's book. So she had this experience of, we should do outreach, which didn't appear to me in the beginning, but then I thought, yes, let's do it.
Definitely.
So she came up with it. Then we also got on board Julia Strahl from. She was working with us here. Maybe I'm sure you remember. Julia, who is very eager to organize things, and she was the perfect buddy in this. And also she is a bit of like an artist, because she's doing pottery in her free time. And that's why we had a chat, and we all figured out we all do something else in our free time that is kind of creative and also requires energy and time, right? Why not display it and use it for something useful, and for instance, for transferring or transmitting the ideas we have about science or the insights we get from our scientific work to the public. And this is how it came about. So we started looking for funding for this exhibit. It took almost a year, I think.
All right.
Then the girls left so they left all the work because, of course, we are all kind of job hopping at this moment.
Yeah, exactly.
But it was really nice because this also taught me just don't give up. Like, stick with people even if they leave. You can do. I think since the pandemic, we know we can just do everything remotely. They came here when we opened the exhibit. Mm-hmm. So we had a collection of photography, mostly underwater and mostly coral reefs, because this is very iconic and it's like it's the best. It gives the best motives to use to present biodiversity that people can really marvel at and that get really engaged with. We really didn't want to miss out on the local things. It's hard to find photographers that really have can present something really nice from the North Sea because it's such a challenging environment, right? Yeah. But we had a friend of mine, Michael Swart. He's also a biologist, marine biologist at Geomar, and a diver. So he actually contributed most of the local biodiversity imagery for our Nordic Seas corner. And an important thing was the art, like an artistic corner. So we had some paintings and comics on the display, all created by scientists in their free time.
Was Mita Mahato? Did she take part in the comics?
No. Yeah, she's also doing. She knows. We talked about it, but she came in, I think, later, and then she was overwhelmed with so many projects and requests. We didn't have her on it. Also, the intention was to have scientists, scientists that produce art. Okay, so all of our artists are scientists.
Okay, right.
By day and artists by night, and it's the collection is still here, so we have it. So to anyone listening to this podcast, we are happy to pass it on. So you can basically rent the photography we have, the artwork. It's up to we need to check in with the artists every time, and we're also in some cases thinking of expanding something. We're planning to have something new in Bremen end of this year probably.
Cool, yeah.
So stay tuned.
Yeah, definitely.
But nothing, like I don't want to give any details yet, and there are no details to be honest. but it's still alive, and we try to keep it alive and even further develop this thing.
Sweet.
So also happy if any of you are interested to do something or join something or come in with your ideas. Just talk to us.
Perfect.
Get in touch. Yeah.
Okay. Yeah. Perhaps more on your personal side or the side of your career. You're from Germany originally, from Bochum, or?
Well, I'm actually Polish.
You are? All right. Roik is.
Well, I think it's a pretty international or not a well-defined. Yeah. People can't identify.
Yeah, exactly.
So, my parents are from Poland. Yeah, and I grew up in near Bochum in Essen, in the Ruhr area, which is like this cluster of huge cities.
Yeah. the
Ruhrgebiet. Yeah, and so I started out as a student in Bochum, which is a huge university.
It is? Okay, right.
It is big. It has I actually stayed there because I thought I didn't really know what to do. And then I started exploring, and they have many faculties. So you could basically study almost everything there, I guess. Yeah.
And you studied biology and English studies?
Yeah, I started out with because people told me when you study biology, you will end up as an unemployed biologist.
Yeah. Okay.
And then I said, okay, I'm going to be a teacher. But then I decided not to be a teacher.
Yeah, okay.
Yeah, pretty quickly, actually. After the first hospitation, I think you call it. Yeah, I just decided for myself, no, I really want to do science.
Okay.
And then I finished both because whatever I start, I want to finish. That's
a good trait.
Maybe I think it's not a good trait.
Maybe not. Okay, perspectives are different.
Yeah. But I stuck with biology.
Yeah. Okay.
And I actually started. Yeah, if you want to know what I started with, it was not marine science. Like everyone was dreaming of marine science. I didn't. I actually was a student, and I enrolled in all these neuroscience programs, or like block course.
Yeah.
You know how so it's like classes and practicals,
Right.
and of course you do the general lectures.
Yeah,
So
okay.
this is it was biology and bio
Right.
sciences, or biotechnology. So it was a mix of classical zoology, but then we had some, at that time they called it genetics, which is dealing with DNA.
Yeah. Okay.
I was into neurosciences, but then I think I don't know why, I just took a diving class. I enrolled, I started joining the dive club at the university. And then I thought I need to do something with my dive license, so I joined the field trip that was offered
All right.
by my then later supervisor for my master thesis. So I went to Egypt with Ralf Tolrean in Bochum.
Okay. Right.
And I kind of integrated well with this group, and indeed so it was really interesting because they were looking for someone looking at the neuronal system of a crustacean. And I said, yeah, why not? Let's do it. So I'm still in touch with the people I worked with in Bochum.
Yeah.
Or at least with one I'm in touch.
Yeah.
That was kind of the window to the ocean. And then I thought, the brain, because the brain was for me like there will be lots of work to be done because we don't know anything about the brain, right? But then I figured out, the ocean, and then you listen to lectures about the deep sea and all these unknowns, and then I thought, okay, there's many unknowns, and I got stuck with these marine, and so my master thesis was actually on a predator-prey system, and it was about plasticity in animals.
Okay. What does that mean, plasticity?
Plasticity means that one genotype would express different phenotypes.
Okay.
So like how you express your genes.
Yeah.
And it was about daphnids, so tiny crustaceans, water fleas, that's the word.
Yeah, okay.
These daphnids are known to change shape. So they morphologically remake when they sense a predator. And my group at that time was interested in what are the pathways and the neuronal pathways, how the sensory input goes, how does it go into this daphnid, and then how does it change when the morphological restructuring is happening. So I did some neuronal stuff on crustaceans. And later on I got interested in this restoration project because I thought, you have this phase of wanting to save the world, maybe. Then and that got me hooked on ecology, because it's like a system, you work with data, you try to connect the dots. So maybe I don't know if I don't know if neuroscience would have been something for me or not, but maybe it's a similar thing. You correlate things and try to understand a black box. Kind of. So
for sure.
Yeah. So, but then I kind of, because by then I had more experience in marine sciences than neurosciences, I got stuck with that.
Nice. And all at the University of Bochum, so that's where you did your master's as well.
Yeah, exactly. And then I decided to go for a PhD at some point.
Yeah, exactly. And let's talk about that. That's so I also started studying marine ecology, or started with coral reef ecology a little bit, and there was always this KAUST. They always talked about KAUST, the King Abdallah University of Science and Technology in Saudi Arabia, and there were always these great opportunities there, but then you kind of had to be satisfied with the idea of living in Saudi Arabia. And so what was that like, studying in Saudi Arabia?
It was nice. I missed the time.
Yeah.
But it's not, at least for me, it didn't feel like a place you will stay long. So that was the nice thing, because most of our jobs in academia are not really permanent.
Yeah.
So at least you go there and you really make the best out of it, and you will leave.
Yeah.
So the mindset is different, and maybe that's why it made the experience really good.
Yeah. Okay.
There's many things about Saudi Arabia which, of course, I wouldn't sign off like and agree with.
Yeah, absolutely.
But so for me, it was the marine sciences why I went there.
Mhm.
I mean, KAUST, so the King Abdullah University of Science and Technology,
Yeah.
is situated just right at the Red Sea.
Yeah.
And I mean, I had before going there, I had the chance to visit for 2 weeks
Mhm.
and check it out.
Nice, yeah.
And then I mean, the place is what the usually a place is what the people around you are, and
Yeah.
I like the people.
Yeah, okay.
So I got stuck with the people, and I went there.
Sweet.
The experience, so Saudi Arabia and like Asia in general. Since I went, I've been in the Caribbean, which is culturally like the total. It's like black and white, right? Total opposite.
Yeah.
And then ending up in Saudi Arabia, I didn't really feel limited there. I was there for the job.
Okay.
But also we were exploring a lot. We went to the desert and
Yeah.
I mean, we talked to people and we had like Saudi friends
Yeah.
there.
Nice.
So because some of them came to study with us. Because in the first place, like when they opened the place, it was more like an American university.
Mhm. So like the campus is quite
it is a
secluded from the city?
compound.
Yeah.
It is a city on its own, so
Okay.
to say. So it's a compound. It's like Oldenburg to Hamburg, maybe like an hour, or is it an hour?
Okay.
Or-
No, maybe a bit more. Maybe.
A bit more, yeah. But
Yeah.
Jeddah, so 70 kilometers.
Okay.
So you would need to go out of. There's walls around
Yeah.
this compound. Outside there's desert, and you can drive to the city, which is Jeddah, which
Okay.
also at the Red Sea.
Yeah.
So you could go into the city, but you were basically living in a compound.
Yeah. Okay.
But the compound, compared to the classical compounds that you find in Jeddah or Riyadh, it's really big. So you don't feel. It's like there's a supermarket. We used to have the only cinema in the kingdom on the
Okay.
campus because there were no cinemas. By now they have cinemas, so the country has
Yeah.
changed
Yeah.
since I left actually, and I'm still in touch with people from KAUST. So things have changed. At that time, I think when I went there was an established community already. Very international, so I spent lots of time with the Latin Americans, with Europeans, Australians, with people from everywhere, basically. My boss was German, by the way.
Yeah, Christian Wulstra?
Yeah, exactly. He's now in Konstanz in southern Germany.
Ah, nice. I didn't know that.
I'm in the north, he's in the south,
Okay.
so far away. I got stuck with a group and with the Red Sea Research Center that I joined. It was also a very international place, and it was very mixed and different research research fields represented.
Okay.
We were on the coral, so we were the Bentic group, and so Chris is a genomics person, so he's really hardcore genomics, and I was more. I used my skills from the restorations project that I was kind of running in the 2 years before to set up some field monitoring sites, because Chris and his group, they were interested in like really working in situ and trying to understand the genomics of these features in the reef. Let's put it that way. Yes, and then the group was also very mixed. So we had like physiologists, we had sequencing technicians, and so I don't want to miss this experience.
Yeah.
It was a good one.
Nice.
But always know at some point it's time to go.
Yeah. And you did go. You went to Kiel, back to Germany.
Yeah, exactly. I went to Kiel.
Yeah, the Christian Albrecht University in Kiel.
I started out there
Yeah.
and joined a microbiology lab to deepen my microbiology skills, because I was an ecologist, right?
Yeah, that's right. Okay.
By then, doing some metabar coding. So I learned a bit about microbiology and some techniques that we can apply. And during this time, I actually wrote my first proposal because I got stuck with corals and the problem that corals have, I think, in my interest in how the microbiome works with the organism that I know best, which was the coral at that time. So I wrote up a proposal, and I was lucky to get it funded, so I could continue my work from, well, I restarted I used my Kaust work as an inspiration for something new. Let's put it that way.
Yeah, nice.
So I moved on from microbial ecology to look more at the holobiont, which is the host and the microbiome together. That's what
okay.
holobiont or meta-organism. Sorry for
So host and microbiome together are the holobiont.
Yeah, so you are a holobiont with all your whatever organismal, like other species attached to you.
Yeah.
So that we speak of a coral holobiont. By now, people speak about the meta-organism. So in Kiel, I was involved in this meta-organism consortium. So I was an affiliated researcher with it's a collaborative research center, right?
Yeah.
It's a DFG-funded research center with a focus on meta-organisms. So which means microbiomes and the host organisms. And from there I could continue working on corals and coral microbiomes, and from there I developed these ideas by meeting all these other people who started working on microbiome manipulation. And I continue this work here at HIFMB.
Okay, no.
Yeah,
nice, definitely. So from Kiel, you from the Christian Albrecht University, you went to the Geomar.
Oh, yeah, right. That was based because the Geomar, it's obviously the Marine Science Institute, and I wrote my grant to be hosted at the Geomar.
Right. Okay.
It was still a kind of in a collaboration with, or I was affiliated to the people from the university, the meta-organism group. And I spent 3 years there, and my major, like this big experiment, was during that time.
Was that the first proposal you wrote?
Yes.
Yeah, your first proposal got funded.
I was so
That's
lucky.
Wild.
I know.
Cool. And you still, but you the funding ran out, but you're still working on the work like it always is.
Yeah, I mean, I applied here with my follow-up project, my follow-up ideas to this.
Yeah.
But there's still some potential and still work to do.
Yeah. Speaking of that, what's next for you then? So when the work is all wrapped up. Wrapping up.
No, I have another plan for, as I said, like the microbiome transplantation has to go on. And so far, I met a couple of people who have done a project in the Philippines, so I'm in touch with them.
Okay.
Let's see what they have found out. I'm really excited.
Are they based in the Philippines?
Yes, yes. I met them just in the conference this year that we had in June.
Yeah.
I have a plan with my colleague who's based in Gießen to have a little, like, this is the follow-up project that is on my mind for 2 years now or so. Yeah. And this will go on for sure. And I myself will need to look for other opportunities to be based at another institute, or find new funding for the new visions that I have.
Yeah, absolutely. I mean, it's very nicely continuing from one step to the next, and you seem to be quite lucky with proposals.
So far.
Yeah.
But you never know, right?
Yeah, exactly. No, I'm sure there will be.
Yeah. So I'm gonna continue. I'm trying to wrap up, but I'm gonna continue a few things that are also in the hands of my collaborators more than in my own. And otherwise, I'm super interested in microbiomes still. Yeah. And not so much in the brain anymore. but I think I will stick around with that for sure.
Nice. Okay, that's perfect. With that, I think we come slowly to a close. But if there's anything that you find we haven't highlighted or that we didn't mention, you can. If you want to bring anything up, you can. Or
not that I like, nothing on my mind now. I think I talked too much already.
No, that's fine.
No, but thanks for this invite.
Yeah, thank you.
I was very happy I was excited and didn't know how this will be.
Yeah.
I'm happy about giving us this platform to talk about our work, because I think that's really important.
Yeah, exactly.
We don't talk about it. Like, especially in such a way.
Happy to hear about it. It was super interesting.
Yeah, thanks for putting time into this. And I love podcasts.
Yeah.
I told you.
Nice. Okay.
Okay, thanks.
Thanks, everyone. Bye-bye.
Want to dive deeper? Surf over to hifmb.de or follow us on Twitter at hifmb_OL.